So with that, I'd like to move on to the final presentation in this session, and I'd like to welcome Professor James Spicer. So this is going to follow on very nicely. Alan alluded to the Checkmate 743 study there, and some of you will have been involved in that study. And it's not surprising that we've got a number of clinical trials available in mesothelioma currently, and many of them are involving immunotherapy. So it's a nice lead-in to James's presentation. I'd also like to thank our sponsor for this session, Novum Law. Our corporate partners have sponsored this session. Professor James Spicer is a Professor of Experimental Cancer Medicine at King's College London, but of course he'll be known to many of you as he practices as a medical oncologist at Guy's Hospital. So welcome, James, and thank you very much for joining us.
Thank you very much, Liz, and thank you, Alan, for a fantastic lead-in. Quite a lot of what I'm going to talk about is relating to immunotherapy, because in mesothelioma, as in many other areas of cancer medicine these days, immunotherapy is beginning to make an impact. I've got quite a lot to cover in the time allowed, but fortunately I think some of my colleagues have been talking quite quickly, and we've got plenty of time to go through quite a lot of data. I make no apologies for showing a lot of data, and that in itself is great news, I think, because we have a lot going on in mesothelioma, and some really interesting trials which are recruiting in the UK at the moment, or have previously read out from patients who've participated in trials recruited in the UK in recent years. Of course, in most recent months, clinical research in pretty much every area except for infectious diseases has had to take a back seat, and that's been a very unfortunate dividend of the COVID crisis. And one of the major lessons that I think the UK National Health Service has learnt from COVID is that if we have to go through some more of this in the epidemic, keeping clinical trials on track and access to new drugs for cancer patients is absolutely vital. And I'm pretty confident that we won't have the sort of research shut down, which for a number of months in the early summer was the case in the UK and in most developed nations, in fact. Anyway, coming to the material that we're going to share together in the next half hour or so, the treatment of mesothelioma is multifaceted, multimodality in many cases, and that's what this trial is here to remind me to tell you. I'm going to be focusing on drug treatment of cancer just because that's what I do. But my surgical colleagues and those who deliver radiotherapy have a significant role to play in this disease as well. And there are interesting trials going on in those areas, which, in the interest of time, I'm not going to cover today. So we're going to start off with some discussion about chemotherapy, just because that remains really the gold standard and front and center of many patients' treatment for this disease. And then we're going to move on to what I've got at the bottom of this slide, the newer therapies, so-called targeted therapy and immunotherapies. So this is rather a dry slide, but I think there are some important themes here, and this is the right starting point from my perspective. I think a lot of people participating in today's webinar will be familiar with this sort of treatment, chemotherapy with two drugs, the one that's really doing most of the heavy lifting, in my opinion, pemetrexed, combined with a platinum-based drug, and there are two of those that we commonly use almost interchangeably, cisplatin and carboplatin. And putting those two together has been a sort of standard treatment for initial treatment for patients with mesothelioma for nearly 2 decades now. And that the result of the trial on which we base that treatment is shown here on the right of this slide. And I think the other thing I wanted to use this slide to do was to introduce this sort of shorthand graphical way of representing the results of oncology trials. and I think it's worth spending a few minutes just sharing how we interpret these sorts of graphs and how they show us whether drugs are useful or not. So this is what's called a survival curve for fairly obvious reasons. I make no apologies for a fairly stark aspect to this presentation. I think everyone participating today is aware that mesothelioma is a difficult disease to treat and catches up with some patients much too soon. And that's really graphically illustrated here. So on the bottom of the slide, we have time, which of course is the precious commodity from a cancer patient's perspective. So this is time in this case until death. This is overall survival, as you can see at the top. And this is probably the most reliable endpoint in trials because, for example, it doesn't rely on rather subjective interpretation of a CT scan. There's no argument about the patient's date of death. And of course, from a patient's perspective, how long one lives is at least one of the most important considerations when undergoing treatment or considering a treatment that might be offered. And then up the side of the curve is a proportion, one at the top, or we might see in some curves 100%. And that in this context is the proportion of patients who are alive at any given time. So you can see from this curve that for mesothelioma, for this group of patients in this trial, and this was quite some time ago now, around half of patients could expect to live for longer than 1 year. And that's where that sort of rule of thumb that some patients are given at diagnosis comes from. But, this is quite historic data now. Anyway, that's an average, and no patient is an average. And towards the right of the curve, you can see there are plenty of patients living for a length of time measured in years, as we now know, in many cases, rather than months. Anyway, that's explaining how these curves work. And then how do we use them to interpret the results of trials? Well, this landmark study, as I say, nearly 20 years ago now, it certainly was recruited 20 years ago, was comparing what since then has been the standard chemotherapy combination, pemetrexed and platinum, with a control arm, and that control arm was a single agent chemotherapy drug, cisplatin in this case. And you can see that there's a separation of the curves. And if you have a mathematical bent, if you don't at all, I've probably lost you already on this particular slide, so don't worry about that. But there's a shift to the right of the patients in the experimental arm, the treatment arm that we were testing in this study. And that means that at any given time, there are more patients alive on the experimental arm of the trial than on the control arm, which of course is a good outcome. And the statistics from this trial suggested that was definitely statistically significant, and with a prolongation of survival of approaching 3 months on average. Some patients do less well than that and some much better. That's clinically significant as well, clinically important to the patients and their doctors. So this became the new standard of care based on this sort of analysis. And the other way of measuring how good or bad a treatment is, Alan's already alluded to, and that's in the bold black letters on the left, and that's this concept of response rate, which we quite often use as a shorthand for how effective cancer treatments are. And particularly for thoracic cancers like mesothelioma, like lung cancer, if a tumor is shrinking, then generally people feel better, they feel less breathless, perhaps their pain improves. And so a response rate for this chemotherapy of above 40% is pretty decent. And even patients who don't respond generally find with this treatment that the cancer stops growing for a time, and that can be associated with improvement of symptoms and quality of life as well. And that's another important readout from many trials, is that we want to know whether patients actually feel better and are more active on treatment. We're not treating their CT scan, we're treating them. So if we give patient loads of toxicities and they live a little bit better, a little bit longer, at the expense of terrible side effects, then we're not really winning. But in this context, with this treatment, a very careful analysis also showed that although we're adding an extra chemotherapy drug, the quality of life was better. So this became firmly the standard of care, and we'll refer back to this standard treatment as we go. But I'm going to move now right into the more present tense by talking about a targeted therapy. And here, if you're not of a mathematical bent, maybe you'll tune into this bit if you're a bit biological, because I'm going to have to explain a little bit of rather fascinating cancer biology in order to understand how some of these targeted treatments work. So as a tumor gets bigger, anything bigger than a sort of a millimeter across, then passive diffusion of nutrients and oxygen into the tumor cells is not reliable. And so for a tumor really to get bigger and cause trouble, it needs to improve its blood supply. And there's a very clever physiology in the way in which it does that. So many tumor cells secrete this molecule called VEGF or VEGF. It stands for vascular endothelial growth factor, but that tongue twister is a bit irrelevant. We call it VEGF for short. And by secreting this peptide, it's almost like a hormone. It acts on the cells lining the blood capillaries, causes them to proliferate, and then as a result, new blood vessels grow into the tumor, which is good for the tumor and, of course, bad for the patient. And actually, this expression of VEGF is higher in mesothelioma than most other solid tumors. So it's an attractive targeted therapy in many diseases, and targeting this biology has been effective in some other more common cancers. And on paper, at least, it looks pretty attractive in mesothelioma because of the common upregulation of this pathway in mesothelioma. So having understood the biology, this is the way cancer discovery and development works, cancer drug discovery. We can now think about drugs to have an impact on this pathway. And there are several. So, for example, there are antibodies. We hear about antibodies, don't we, endlessly in the COVID pandemic, because antibodies are a naturally occurring part of our immune system, and they're able to bind specifically to all sorts of targets, and if it's a virus or a bacteria, neutralize them. And we can use genetic engineering technology to manufacture antibody protein drugs, which will recognize anything we like, and we can design antibodies to recognize VEGF. And such an antibody is called bevacizumab or Avastin, and we'll talk about that in a minute. Another way we can interact with this biology is by using more conventional small molecule pharmaceuticals, and these are a bit more like the same size as a chemotherapy drug. And these sort of targeted therapies have been quite useful in lots of other cancers, and a number have been designed to actually hit the receptor for VEGF. And many of those have been tested, and we conducted a trial of one of these drugs at Guy's, in fact, a few years ago, but without very much success really across the class. So we're not pursuing those small molecules anymore, and I will concentrate on these antibodies targeting this process, which is called angiogenesis. So that's from the Greek, isn't it? I'm not classically educated, but I think that just means the generation of blood vessels. Angiogenesis is the process we're targeting, and so bevacizumab, this antibody, is an anti-angiogenic targeted therapy in the jargon. So this slide shows the design of a trial which was conducted mostly in France to test whether this antibody added to the standard chemotherapy that we've already discussed, whether that improves outcomes. So to the uninitiated, this is a rather complicated slide too. What are all these boxes? Well, this is to introduce the concept of randomization in a trial. So quite a lot of what I do is testing very new drugs in phase 1 trials where we're just seeing whether the drug is safe. If we really want to know whether the drug is effective, we need to have something to compare our experimental treatment with. And so we have this concept of randomization in a trial. And you can see on the left-hand side of this trial an abbreviated list of what we need for a patient to be eligible for this study. Broadly speaking, a fit patient with confirmed pleural mesothelioma. And then the computer randomizes them, if they agree, to either the standard treatment, pemetrexed plus platinum, which you're familiar with, or the experimental treatment in this therapy, and that was just exactly the same chemotherapy plus the bevacizumab antibody. So you can see it's an ethical trial, and it's important to design trials thinking about, what the opportunity cost might be for the patient. If we're testing an experimental drug and we're not sure that it's helpful, we need to make sure that we're not disadvantaging the patient if the trial turns out to be a failure and the drug is not helpful. So here you can see there's no downside really for the patient from the point of view of effectiveness, because everyone's getting the chemotherapy as standard, and then some patients are getting the experimental drug which might help. We don't know until we do the trial. So this study has actually been completed now, and here's another survival curve which your experts are interpreting now. But you have to be an expert really to interpret this curve, I think, because the two curves are pretty close together, aren't they? Some of the numbers show that there is a statistically significant separation, but how clinically significant that is, with an average survival separation of just a couple of months on average, whether that's really a difference which is going to be important to patients and noticeable in an individual patient, is debatable. And certainly adding this targeted therapy makes the chemotherapy more toxic, something I haven't got time to go into in detail just at the minute. So what do we do with this result? Well, it's been differently interpreted, and this is very common in oncology around the world when we have a result which is a bit equivocal, a bit not quite sure what this means. So, for example, in the NHS, this drug is not routinely available. It is an expensive drug and it's not routinely funded together with chemotherapy. So chemotherapy alone remains the standard treatment outside of a trial. And I think that's not something that me and most of my patients lose too much sleep over, because of the caveats that I've already talked about. But in some parts of the world, particularly in France, where they did this trial, that's just the nature of medical science, I suppose, it has moved the needle, and for many, the addition of bevacizumab ought to be considered a standard in this sort of context. And so for the next trial I'm going to tell you about, the standard of treatment, the control arm. Here's this randomization diagram again. At the top, the control arm at the top is a new standard, that is chemotherapy plus bevacizumab, and you can see on the experimental part of the trial at the bottom which patients are allocated to 1-to-1 at random again. And you can see that bevacizumab and chemotherapy are present as an insurance, if you like. So what's the trial about? It's about the addition of this new drug. So now we're going to shift gears into the final chapter, although the longest one of my talk, and that's this drug called atezolizumab. All of these long names you shouldn't commit to memory. In many respects, I think drug companies invent almost unpronounceable generic names, so that what we're forced to do in the end is use their catchy brand name, which is almost always, funnily enough, easier to say. But I'm a bit of a stickler for generic names, so I will call this one atezolizumab. And the mab bit at the end is helpful anyway, because it reminds us that it's a monoclonal antibody. So this is a protein-based drug based on an antibody, just like bevacizumab, but now we're targeting these immune aspects, which you've had very nicely introduced to you by Alan. So a little bit like the anti-angiogenic drugs, which are targeting something a little bit one removed from the tumor. With immunotherapy in cancer, again, we're unlike chemotherapy, where we're aiming straight for the cancer cell bullseye. Here we're really approaching the cancer a little bit tangentially, because what we're seeking to do is to boost the immune system and then allowing that to do our anti-cancer job for us. We're not directly addressing the cancer cell. So there's quite a lot of science in this slide, which I think for the purposes of today is not really very helpful. But it helps me to talk about some of these things, and Alan's introduction, I think this makes this much more approachable. In rather sickly green at the top in this diagram is a T cell, and the T cells are, if you like, the foot soldiers, or the stormtroopers even. They're the ones who really do the dirty work of the immune system. And they're able to recognize abnormal proteins which are not meant to be in the body, highlighted in red in the middle of this diagram. They're able to recognize these foreign proteins, and provided the T cell gets all the right signals, it will unleash its warheads to kill the target cell. That target cell might be a lung cell infected by coronavirus, for example. And we're beginning to think that T-cell immunity is very important in coronavirus disease. But in the context of a cancer talk, the abnormal protein we're talking about is on the surface of a tumor cell. A tumor cell, by definition, is different from normal tissues. Otherwise, it wouldn't be a tumor cell. And the difficulty from the tumor's perspective is that it needs to express some abnormal proteins to allow it to grow abnormally. But at the same time, it needs to avoid these patrolling T cells, which are just looking for abnormal targets to kill. And one of the ways in which they push T cells away is with this target that you've heard about already from Alan, which is PD-1. And what that stands for, it doesn't help me understand what it does, so I'll just call it by its acronym, PD-1. And that's this little green-shaped receptor, lock-shaped thing in this diagram. And tumor cells have evolved, and the reason they're successful tumors, unsuccessful from the patient's point of view, is because they express a lot of this protein PD-1 ligand, which binds to PD-1 and switches off T cells. So this gives us a great target for immune therapy. And this break in Alan's analogy, we're able to cut the cables to by developing these immunotherapy antibodies, which bind to PD-1 and stop this fending off that tumor cells are usually able to do. And that's the theory, and the practice in the laboratory experiments done, I don't know, 10 years ago now, were pretty impressive. And what was really impressive is how active some of these drugs are in some human cancers. And you've heard from a melanoma doctor who was really in at the birth of all of this, how in some diseases these drugs have been really practice changing. Lung cancer is another one where we use these sorts of drugs in most patients. They don't help all patients, and we're still working on that aspect. But the question for today is, are these therapies helpful in mesothelioma? Because mesothelioma is a relatively cold tumor, as you've heard, and can be a bit difficult to treat with immunotherapies on paper. Well, in the first instance, we look at the results of kind of trials that I do, phase 1, phase 2 trials. So these are not randomized trials. We're just testing the drug to see whether they're safe and whether they're associated with a response. That's really what we can do if we don't have a control arm, is to see how many patients have their disease shrinking with these drugs. And the text is probably getting a bit small for your screen, but for some of these antibodies, you can see the response rate, RR, for pembrolizumab and nivolumab, two very widely used drugs elsewhere in other cancer clinics, is about 25%, which is not fantastic, but it's of interest and certainly worth further investigation. For another drug, in fact, chronologically, the very first one to be tested in mesothelioma, even harder to pronounce, tremelimumab. This targets another protein a bit like PD-1, but distinct from it. You can see the response rate there was really quite modest, below 10%, although in a tiny trial of only 29 patients. So with that sort of background, moving into the last lap of my talk, I want to share with you some of the results of the very first randomized trials to be conducted in mesothelioma. And these are the first, with a randomized trial, we can now really start drawing conclusions about whether these treatments are helpful. So I'm going to talk about some trials which are actively recruiting, which I'm sure will be of interest to you in a moment. But again, to bring us up to speed and fill in the context of how effective these so-called checkpoint inhibitor immunotherapies are, we need to look at some of the recent randomized trials. In fact, there are so few in mesothelioma that even in the time allowed, we can cover most of these. So this was the very first randomized trial of an immune checkpoint inhibitor in mesothelioma, and it was conducted in patients who'd had chemotherapy before, so-called second or third line setting, and they were randomized either to this tremelimumab antibody, which I just mentioned, or to placebo, and that was felt to be ethical at the time because there was no standard of treatment for patients who had chemotherapy already. Here's the survival curve, which is a bit small, but I don't need you to look at it very closely because those two curves are completely superimposable, and therefore, unfortunately, there was really no difference. There's no contribution to this immunotherapy over and above placebo. That's why control arms are so vital in trials, is that without the placebo control, we wouldn't be able to interpret whether this was a good survival curve or a bad survival curve. As it turns out, it's no different from if we offered the patient observation and supportive care alone. So that was really quite a setback and disappointing. You'll see in the bottom right, there's a reference there, and Lancet Oncology is quite a respected high-impact journal, and I think that's an important point to note. Nowadays, in the old days, negative trials have quite often got buried, particularly if pharmaceutical companies had anything to do with it. And now, I think it's an absolutely vital ethical principle and something to be proud of in the oncology community. We're just as interested in negative trials, and they get published in just as high impact journals as the positive studies which turn up on the news at 10. So a negative trial, but a really important result and something to learn from. Since then, there have been some randomized trials of what generally are acknowledged to be more potent immunotherapy drugs, ones like pembrolizumab. This was an important trial to the UK, although it was a pan-European study. It was a study led by my colleague Sanjay Popat at the Marsden, and we recruited a lot of patients at Guy's. And this was the randomization, very similar to the previous study I just showed you, the same population, patients, in fact, who just had one line of treatment before. In this context, by the time we did this trial, we felt that vinorelbine, another chemotherapy drug, was a standard of care in the second line setting. And the randomization was to pembrolizumab, this immunotherapy drug. And importantly, we allowed crossover in this trial. So if patients' disease got worse despite the chemotherapy, they were allowed then to switch to the immunotherapy, so that the patient and their doctors felt they had access to the immunotherapy drug either way. And we were able to draw a conclusion from how long patients' disease remained controlled before they had to switch. So what was the result of that trial? Well, we're getting right up to date now because this was presented very recently. And this is the outcome, the primary outcome, which was a little bit disappointing. You can see that these curves overlay each other completely. This is the time to progression, and that's why this curve is a bit wobbly, because the time at which patients progress rather depends on when we do their CT scan. So superficially a bit disappointing, but of course the control arm here was not placebo, and so if you're a glass half full sort of person, you might say, well, this is a way of avoiding second line chemotherapy and having something which doesn't have the side effects of chemotherapy, namely immunotherapy. Now, immunotherapy does have side effects, and we may have time to talk about that, and some of them sometimes can be quite severe, but in general, immunotherapies are better tolerated than chemo. So patients who can access immunotherapy instead of chemotherapy often choose to do that if they can. One of the silver linings of the COVID pandemic in the mesothelioma context has been that we've been allowed to use nivolumab, which is a drug a bit like pembrolizumab, instead of vinorelbine in this context. The rationale probably being there's never been a randomized trial. The rationale presumably being that we're avoiding the toxicities of chemotherapy, in particular the immunosuppression of chemotherapy, just in the context of COVID pandemic, when we don't want patients being more vulnerable to infection. I'm relying on Liz to start making faces at me if we're running out of time, and she's looking quite calm at the moment. But I am going to accelerate a little bit now and talk about a study just reported a few months ago, which I think really moves the needle in the way we understand these therapies in mesothelioma. There's a lot on this slide, and it's, I don't want to labour the point. This is yet another way of presenting the results of a trial. And if you were all oncology trainees, I'd take you through it painstakingly, because it's an important way of presenting the results of a small trial. But we'll try and go through this briefly, so forgive me if it's a bit superficial. The way we present the results in these sorts of trials is that each of these bars is a representation of how patients' tumours shrink or grow, and a downward deflection is good in this context. That's a shrinkage. And there are a couple of different arms of this trial. This trial was called MAPS-2, so you can probably guess it was done by the French again. And they were testing in a non-randomized way either nivolumab on the left, or the combination of two immunotherapy drugs, nivolumab and ipilimumab. For the sake of saving the twisting tongue, we call that one Nivo-Ipi, or even Ipi-Nivo. And that combination is very widely used in melanoma, and in melanoma is used at quite high doses, particularly of ipilimumab, and can be quite toxic. But at slightly attenuated doses, it's also active in mesothelioma. As you can see, generally speaking, this curve there's more downward deflection on the right than on the left of these two plots. And so this was used as the basis to design a trial testing Nivo plus Ipi in the first line setting. That is patients who'd never had any drug treatment for their mesothelioma yet. And this is the so-called Checkmate 743 trial. Checkmate is a sort of acronym which Bristol Myers Squibb, a big pharmaceutical company, used for their trials testing nivolumab. And the 743 is an indication of quite how many trials there've been across the whole world of oncology using these drugs. And the arrival of these randomized studies in mesothelioma, if you're still with me on this talk, has been relatively late in the story, just because mesothelioma in the minds of many in pharma is regarded as a less common disease and presumably a less prominent market for them. Anyway, this trial has now been conducted, and it's been well conducted and has an informative result. This is the randomization, and again, you're experts at interpreting study design now, so there's nothing new here. Actually, the same first-line population of patients with mesothelioma. They need to be fit. That's what PS means, performance status, and no prior chemotherapy. And the randomization was an ambitious one, ambitious in the sense that, unlike the other first-line trial that I showed you, the experimental arm on this trial, patients received just the two immunotherapy drugs. So this is a head-to-head comparison of two immunotherapy drugs against the standard two chemotherapy drugs. I don't think this projects very well. The bright green box is the standard pemetrexed plus platinum head-to-head comparison. So how did that trial go? Well, this was the primary outcome of the trial, and again, this is a survival curve. You can see now that the months along the bottom of the study are getting up to some quite high numbers, and although the interpretation of this trial, this was presented by our Dutch colleague Paul Baars at the World Lung Meeting, virtual lung meeting, of course. No oncology meetings have taken place in person this year for obvious reasons. But this data was reported in the high summer of this year, so just a few weeks ago. And here's the primary outcome, and you can see the experimental arm, which is in the orange, is doing better than the chemotherapy superficially. That's the take-home message, and the numbers, which you probably won't be able to read, are the statistics, and the statistics are definitely significant. So how do we interpret this result, and should this now be the new standard of care in immunotherapy first line? Well, this is a really important so-called subgroup analysis. So I've just showed you the bottom line, which is the whole population in the trial, randomized and looking at the two arms. Here we divide the two groups into the two important subsets of mesothelioma, which many of you may be familiar with. By far the most common is the epithelioid sort of mesothelioma. An important but much smaller subgroup is it consists mostly of something called sarcomatoid mesothelioma, and these tumours have actually quite look quite bland down the microscope and rather few immune cells, and the stroma is really rather plain, and they do look quite different from the more common epithelioid mesothelioma. But importantly, you can see even from a distance that these two sets of curves are quite different. I think it's important to note that the immune therapy arm does pretty much the same in the two groups. You can't read the numbers there perhaps, but the proportion of patients alive and well at 2 years was around 40% in both groups on the experimental arm of the trial. But you can see the grey curve, chemotherapy, that performs a lot less well in the patients with the sarcomatoid mesothelioma, and that's something we've been aware of and cautious about in mesothelioma oncology for some time, that treating sarcomatoid mesothelioma can be quite hard work. So it may be that there's a specific focus for this sort of therapy based on this one trial in patients who have the non-epithelioid sort of tumour. And you can see in the more common form of mesothelioma on the right, the difference between the two treatment arms is really rather minimal. And what do we... I'll wind up now, thank you. And what do we do with that information? We need to, I think, lay some caveats on top of this. On the left here, we've got we have response rates which actually show that the response rate with the chemotherapy is higher than that for the immunotherapy, and this is not a survival curve. This is a time to progression sort of curve, and you can see that the experimental immunotherapy arm in orange drops off rather alarmingly compared with chemotherapy. So there is a fear that there's a group of patients who don't respond very well to immunotherapy, and that they may be better off having chemotherapy in the first instance. So all of these details are to be worked out before combination immunotherapy with some quite significant toxicities, which I won't go through on this next trial. Suffice to say that the toxicities of the immunotherapy combination were at least as significant as chemotherapy. We have work to do. This is a complex slide, which I think in the interest of time I will skip over. Suffice to say that this rather alarming cartoon on the right just shows that the targets that Alan and I have shared with you today are just scratching the surface of how complex biology is. But that's good, because we have drugs that have some activity and lots of other unexploited targets. But some of them are difficult to exploit, and this slide summarizes a trial of a new immunotherapy drug that we tested at Guy's, and out of 12 patients with mesothelioma, we saw no responses at all. So we have to take the rough with the smooth in this world. And finally, Liz, I couldn't talk about trials in mesothelioma without mentioning CAR T cells, chimeric antigen receptor recombinant T cells. This is quite complex and potentially toxic treatment, but seems to be arriving in solid tumor oncology, and mesothelioma has participated from the beginning with this trial, which you can read about online because it was reported last year at the American Association of Cancer Research. This is about taking T cells from the patient's circulation, retargeting them against the tumor, and then injecting them into the pleural space. And this diagram shows you reminds you what the pleural space is around the lung inside the chest, and we can access that with a small tube that patients walk around with quite comfortably. And I introduced that because on the back of this quite successful study, just opening in the UK, is this trial of delivering a virus called adenovirus, which has been doctored so that it doesn't divide anymore, but it does express an immune molecule called interferon alpha, a so-called cytokine, which can boost the patient's response to all sorts of treatment, including chemotherapy. And in this trial, patients have an intrapleural administration of this virus if they're on the experimental arm of the trial, represented here. So this study is going to be recruiting at Marsden and Guy's in London, and about 3 or 4 other centres in the UK, and it's an international study just sort of winding up now, winding up in the sense of beginning now. So I'll finish there. I hope I've emphasized that chemotherapy, although it's been around for a long time in this disease, is extremely effective. And the new in the vanguard now is both targeted therapy, which seems to be having some impact in mesothelioma, and more recently the immune therapies are where the trials are going. Whether the results of trials to date are enough to change our standard of care is another conversation, but as I've highlighted, one of those drugs, nivolumab, is currently available in the UK for some mesothelioma patients, strictly in the second line, on the back of the NHS's special COVID prescribing arrangements. Whether we'll retain that forever, I don't know, and probably depends on the results of ongoing and future studies. I'll finish there, Liz, and thank you all for listening.